So thank you very much for your attention. We're going to try to contain a multitude of information today in a very short amount of time of a very interesting topic. We're going to talk about some biosemiotics in microbiome studies. And I just wanted to share with you first my excitement, not only of being here, but that in genetic research right now has allowed that we can actually describe a host with all its microbes and the metabolites. And this actually has a huge implication, not only for an individual, but as a society, we actually have some interesting results. So the good thing about this is that, well, I don't know if it's good or bad, but we have high expectations about these results, not only in in research as a scientist, but we also, the general public, actually expects a lot of what their microbes can do to their health. Actually, there's a lot of information out there. You can buy your probiotics on the supermarket, but actually this has huge implications that we have to look around. So even though we have all this information, there are some questions that still are not answered. And I would love to see what the biosemioticians have to say about this. But we know that we are composed by, we have these microbes in us, but actually what are they doing? How are they doing it? And what does it mean to us and to me as an individual? So first we're going to start with some concepts. And we know we define a microbiota as the microbes that are part of a metazoan host. A microbiome is not only the microbes, but also the genes of these microbes. We have to also state that most time when we talk about microbiome, we are referring a little bit more to bacteria, but actually we have bacteria, fungi, viruses, and archaea, and we can study each of these individually. But most of the studies are on bacteria. When we talk about metagenome, it's the collection of all these genomes of the microorganisms that I have just mentioned. And a holobiont is a host with its symbionts. When we talk about hologenome, we're talking about the genomics of the host and the genes of the microorganisms. So saying that, I just wanted to emphasize in some points. I'm not going to go through all this. I just wanted to let you know, we have several different quantities of microbial species in us. They are different in each part of the body, and most of it is in the colon. So just some numbers, right? So we have 30 trillion human cells, but we have 40 trillion bacterial cells. So that's a lot. And in the case of the genes, we have 22,000 estimated human genes, but we have 2 million bacterial genes only in the gut. So actually we are 90% microbial and 10% human. So that's the question I want to ask the biosemioticians. What do you think about all this? This has a lot of implications. And even in an adult life, the microbiome is mostly stable. There are several factors that can influence the microbiome of a person during their life. There's a lot of controversy right now in when does this microbes start actually they begin with a person. Is it before birth or during birth? So the birth delivery is one of the factors that we know that influence the microbiome. It's not the same being birthed by vaginal delivery or cesarean. Also, age, it's not the same, the microbiome of a newborn or the elderly. When I say drug use, mostly antibiotics, this has a huge influence in the microbiome. Of course, environment, because it's not the same living in a big city or in rural areas. And the diet, of course. And with this, I just want to make a quick commercial. Please do not forget always your vegetables, your fruits, but especially your fiber. If you don't eat fiber, your microbes are going to suffer a little bit, so fiber is crucial. I'm not going to go into all this, but there's a lot of research going on about how microbes influence our health in general. There are some diseases that are already linked very well to microbiome. We have a lot of interest more in the gut-brain microbiome relationship, going through the immune system, that this has a lot of layers of communication, so it would be interesting also for more biosemioticians. And all of this also comes with some challenges. We have some challenges in the biomedical sciences when working with this, because we're getting a lot of results. We don't know what to do with them, or we still are interpreting not as good as we should. So first of all, getting a good sample is complicated, because as I said, we have several microbes all around the body. How do we get the ones we are actually interested in studying? Data, of course, because there's a lot of information that we need to process. Animal models, it's interesting because we are making conclusions about results in studies that actually use animals as models because of bioethical reasons, but what does this imply for individuality, right? There's something that we have to go inside there. Microbiome diversity, of course, because there are several parts of the body. I'm referring mostly, I didn't say that at the beginning, but to the human, right? To the human microbiome, because we know that microbes are everywhere, and we can talk also to specific microbiomes depending on the host. And also correlation versus causation. This is very interesting because sometimes when we get the data in the labs, they're going to conclusions too radically, and we cannot go and say, as I said before, that the presence of one or other microbe is going to cause this. We infer a little bit more, and it's more like we have to link only the presence to the microbes. So we have more unknowns than knowns during this studies. And I wanted just to bring this so you can see, this is a sample from my lab. Actually, I don't want to gross anyone out. That's my sample, my fecal sample. That's my result. I didn't want to bring like a graph. We did this workup. So just for you to see the amount of species that you can find there, the ones with the capital letters, the bigger ones are in higher quantities. And just to show you that, yeah, I contain multitudes. In this phrase, you're going to find it in a lot of places, not only in pop culture, but we like it more the poetic part by Walt Whitman. So that's a brief introduction, and now Óscar is going to talk about all the biosemiotics that I think you're going to find more interesting than all this information. So.
Thank you. Thank you. Yeah. Thank you, Raquel. Friends, colegas, thank you for all. I want to focus the biosemiotic notes of Holobiont about these three issues and a bit conclusions. The idea, the swarms, starting but in Holobiont concept, has this homeomorphic equivalent in just pair of major terms on swarms. And I want to use this topic, algebra topic, and the homeomorphic equivalence that occurs with concepts that even have different forms, attributes of functions applied in differentiated environments, take certain semantic similarities or equivalence in the functional morphological type, leading to a significant link between them, that they are equivalent implies that they are also homologous. But it's go beyond of the concept of analogy. Is more than analogy, this concept. Is not a metaphor also. Semiotic competence is delegated to decentralized units such as swarms, while Hofmeier reflected on the decentralized cells that swarm in the host organisms. We try to develop this concept in the set of microbiota that, for the most part, reside as symbionts in the digestive system and more specifically in the intestines. Our problem lies in the question of the Holobiont has a meteorological and pre-individual system. Can we say that the Holobiont is a world of the single sum of the parts? Everything will depend on the future approaches to the functional and metabolic network of the microbiome as an agency that interacts with higher hierarchies of cells. Lin Hugh and Scott Hilbert remind us that eukaryotic organisms, and specifically human beings, are Holobiont systems integrated evolutionarily and anatomically, immunologically, and physiologically, as well as our development, united through mutual trust, like development scaffolding and mutual construction, and as a niche construction. The multi-level semiosis generated between the species of microbes and between microbes and nervous cells and biofilms inside the intestinal rugosities makes up a new development of functional individuation. It turns out to be essential to understand better the transfer of information, called transductive operation, between symbionts and integral parts of the hierarchical swarms of the human host, as well as their role in the development of mereology, agency, and even consciousness or proprioception, as the dichotomy between body and mind. The psychic mode of individuation is inseparably coupled to the pre-individuation of the Holobiont, and we have to think of a united psycho-collective mode of individuation. We transform psychic individuals into subjects and the Holobiont into trans-individual. The Holobiont collective can generate an affection, Simondonian affection, it's said, with a certain group of hosts in a dynamic of exogenous, endogenous transfer on certain microbes. We can think of a situation of collective stress where an individual's microbiota can be modified by a set of control and psychosomatic metastability. It can enter a phase of inter-individual resonance where the group of hosts can be affected by each other, generating organic dysfunctions and pathological infections in populations. It suggests future research on the notion of the collective affective individuation in population immunology. Who will also say that the Holobiont integrates the viruses that host our body, lungs, gut, etc., with microbes? In this case, studies on immunology and epidemiology open up a new biosemiotic perspective from the biocommunication. Interspecies will extend the concept of individuation beyond the hierarchical model of encapsulation that is still a part of mereological world. Simon Dunn's new method of alagmatic epistemology, an exchange of information between analytical science and analogical science, could offer this new ontological and biological paradigm of the living being and, above all, of the concept of the human being and their populations. Simon Dunn's transductive operations, continuums, and their interplay with swarms on swarms offer a new perspective on Hofmeyer's and biosemiosphere through the study on the ontogenetic pre-individuation of the Holobiont. Why with the individuation issue, the mereology entangles on the ontology process of living being agency. The problem of unveiled boundaries persists from the Holobiont perspective. When the microbiome swarm establishing the topological zones or places of niche, we have a fuzzy zone in the body, with microbiome unwelten and microbiome inunwelten developed. There are sub-unwells and sub-inunwell zones that require focusing on biosemiotic perspective to understand what's happening in the processes and how to say these biotopes. Merology implies that the whole is more than the sum of the parts. But it's not easy to deliberate what is the total sum of parts of hologenomes and microbiomes that are not part of the holobiont. We want to consider the mereotopology as a part of the answer that needs to study deeply. Mereotopology is built out of mereology with a topological component, thereby allowing the formulation of ontological laws about the boundaries and interiors of walls, relations and connectedness to the concept of the surface, point, neighborhood, and so on. It is necessary to include not only what it means on an entity to be composed of parts, but also what it means for parts and walls to be continuous or discontinuous in Holobiont. Whitehead considered region rather than point as a basis of through point-free geometry in the Principia Mathematica, and logical and ontological conception with contiguity and connection in the book of Process and Reality. Currently, Gerla and Miranda develop a logical process of inclusion based in point-free geometry that implies the conception of infix, which corresponds to the binary parthood relation, a standard feature in mereological theories. Don't forget that mereology is defined as the calculus of the individuals. Conclusions. The concept of the holobiont is revolutionizing biology thanks to the awareness of the microbiome, the virome, and the microbiome that constitute the sum of the parts of the microbiota. It is a question that leads in the acceptance of the holobiont as an ontological entity in biology that surpasses and previously envisaged conception of the individuation. Anton Marcos and Hanna Zvorkova expose in chapter 10, Epigenetic Processes and Evolution in Life, a very exciting idea regarding that the holobiont encompasses the concept of the multi-level selection theory, which argues that units of selection can function simultaneously and that across various levels of biological organization. Davis Sloan Wilson and Elliot Solberg used the example of Matryoshka dolls as multi-level selection models. Also, the example is valuable insofar as it may seem somewhat compact, and dynamic idea on a dynamic multi-level swarm is not insignificant, to which Hofmeier took advantage of a better approach, and which can bring us even closer to a dynamic mereotopology of the holobiont, with its bubbling umwelt, inside out of sight, the body of the organism. It's a good example of transindividuation. We can also be a fluctuating chimera, as Hugh and Gilbert say, but the truth is that this chimera is still a reflection of our identity as a subject. We are clear that the microbiome both helps and harms the production of neurotransmitters and their signaling, affecting psychic aspects, go far beyond the decision making about the food I think I want to eat. Who decides to take a carrot or a yogurt? Me or my microbiota? But how does the microbiome affect the ability of the astrocytes to generate these other neural connections? As if Ortega said, paraphrasing your school, I am myself and my circumstances. What kind of circumstances exist when I recognize that my symbionts allow me to stay alive or die? Or be me? One last note to finish. When I see a plan, what do I understand for it? František Baluska and Stefano Mancuso have observed that auxin behaves like a neuron, transferring signals that allow the others of the adaptive behavior beyond the interaction of multitropism to escape through the roots. and that both Charles and Francis Darwin already pointed out power of movements in plants that Kalevi explained the last Sunday. That auxin is related to the sensory motor circuit of the plants. We not also know that the certain bacteria, such as Rhizosphere, PGPB, the plant growth promoting bacteria, are beneficial for auxin production and root and branch growth. Well, so thank you very much.